Hi everyone, this is technically our first lab experiment in 103, Biology in Everyday Life. It's about chemical aspect. In the theory part, it is, I think, chapter 3. Now, as chemical aspect, it has another name that we call it as organic molecule. The reason we call it organic molecule is because it contains carbon in the building basic block, and this is why we call it organic molecules. Also, we call it as large molecules as well. Now, it has three different names: chemical aspects, organic molecules, and large molecules. The reason we call it large molecules because we need it in large quantities. We need it as a humans on a daily basis, whether we get it from food, from vitamins, supplements. We need it in large quantities. Now, usually, I explained over here in the soft copy that I will send what is organic compounds. Now, if I call it organic compounds or large molecules or chemical aspects, it's the same name. It contains carbon, atom linked together to form whether a chain or a ring. It is separated into four main classifications. First, carbohydrates. As you all know, carbohydrates, also it is separated into three, depends on the molecule, the sugar molecule attached. If it's one sugar molecule attached to the chain, it's called monosaccharides, disaccharides if it's two, and polysaccharides if it's three. So carbohydrates, the first classification, it is also divided into three main sub classification, let's say. Monosaccharides, as an example of it, is glucose, the one that we're using to sweeten our tea. Disaccharides such as sucrose or lactose we found in dairy products. Polysaccharides, we're speaking about starch or glycogen. The second classification, before we go to the second classification, this is the test for carbohydrates. Usually I use a Benedict reagent, which I will show you the procedure in a minute. Benedict reagent, it's a reagent that reacts with the sugar found in any food items that I'm looking for. It is made of copper ions, basically 2 ions of copper. If I add the Benedict reagent to any food items and I reduce it, meaning by heating the reagent itself with the solution that I'm looking for, the 2 ions of copper will be reduced into 1. Now, reducing heat, I will just keep it in a hot water bath. I won't heat it direct to the fire, and this is enough. The reduction with the percents of sugar will change the color to a brand new color. It is a brick red, and if you keep that brick red standing for 1 hour, it will precipitate in the bottom of the test tube. So Benedict for carbohydrates, basically reducing sugar to be more specific, and it is this is the definition for Benedict reagent. It's very important to know the definition. This is exactly what we will be doing, and this is the brick red we are aiming for. Iodine for starch. Starch is the sub-classification for carbohydrates. But if I'm gonna look for starch, I won't use Benedict reagent. I'm gonna use Lugule. This is the Latin or scientific name for iodine. Now, the iodine, we find it in all pharmacies. We use it as a disinfectant. So this is the regular iodine, or pitadine, if you know the name of pitadine. This is the one I'm using. If I add iodine to a piece of bread, similar wise, it will change the color from iodine's color, which is orange or brownie coloration, into a black blue coloration. In our test, we will use a starch solution, and this is the black blue we're looking for. Proteins is the second classification. If amino acid is attached to the chain of chemical aspect, not sugar, amino acid, then I will call it as a protein. And what binds them all together is very unique bond. It's called polypeptide chain. Burette reagent is the reagent I'm using to test for any food items that contain protein. It's very simple. Once I add burette to any liquid that contains protein, it will change the color to purple coloration. Very direct. I don't need to heat. I don't need to reduce it. Instant color change. Lipids, as we all know, an example of lipids is oil. This is a take-home experiment, which I will explain in a minute. Now, this is the procedure, which I will do it, and this is where you will get to put or apply your results. You will end up writing a lab report. Your purpose is testing food samples for the percents of large molecules, that is carbohydrate, starch, etc. This is originally made for on-site students, so it is not a group of 4, it's individual. Okay? Now, as you can see over here, before we start how to write a lab report, over here there is always a test tube where actually it has only water, and when I mix water with the reagent, it only gives me the color of the reagent, nothing else. This is a negative control. It's very important in any biology experiment that we use a negative control and positive control. This is a positive control. Why am I required to do that? It is very important to have negative and positive control to compare my result to, for comparison. Now, why do I have to compare? To have a precise and accurate result. Very simple. Okay? So, going back, to our lab report, it's individual lab report. Your purpose is testing samples of food for the percents of sugar, starch, lipids, and proteins. You will type it and you will send it to me. Don't worry about the assignment. I will clarify it in my email, what to do exactly and where to send it, whether Moodle or to my email, through the email that I am sending to you. That's all. Now we will go for the practical part. So this is mainly the practical. As you can see, this is a Clifton plate. It's a heater. This is the beaker where I keep the water, to heat the water, to use it as my reduction, the heating part. Over here, as you can see, I have 3 test tubes, glucose solution, starch solution, it is labeled, egg white, and I have my negative control, the water. So I will add Benedict reagent. This is Benedict reagent. It's a blue colored. I will add it to all 3 test tubes and I will apply it over here, okay, for reduction. I hope it is clear. I don't have enough time for my video and to picture everything. Now, why do I have to add Benedict to all 3 of them? I need to know for sure that starch or egg white doesn't contain sugar. So Benedict reagent, it is made to test for the percents of sugar. I will keep it for let's say 5 to 7 minutes and I will come back for the result. I just need you to see the start, how it start to change in color. It becomes yellowish into a greenish, let's say, and I will keep it till we have the color that we're aiming for. It's very important. This is the reason I should use always a negative and positive control. Look at the starch solution, and this is egg white. Nothing happened so far. Now I need you to look at the glucose. Look how it becomes a coloration, reddish into yellowish, and this is why we call it brick red coloration, because it is in between the colors. And if I will keep it standing for 1 hour, that redness will precipitate in the bottom of tested tube, which I will show it for you after 1 hour standing. Okay, so wait for the result till then. But also I need you to look at the negative control. Usually we keep them together to compare our result. Look at the starch, nothing happened. And if you look at egg white, also this is not the aimed color we're looking for. We're looking for brick red, and it will precipitate after 1 hour. So we will wait for that till it precipitates. Now for our second experiment for starch testing. Now to test for starch, which is the second sub-classification of carbohydrate, as I explained earlier, we don't use the same reagent for sugar. We use another reagent. It's the iodine over here. Okay, it's browny yellowish in coloration. When we add it to any substance that contain or liquid contain starch, it will change into a black blue coloration. Look at that. So why we call it black blue? Because when you look at it, it is light black, so we call it black blue, or you just refer it as a black coloration. Okay, so over here I have also glucose, starch, egg white, and my negative control, water. So I will add a full withdrawal. Now full withdrawal, it's almost 1 milli, which actually equals the 1 milli over here. We have to work in an accurate and precise measurements to have an accurate result. So 1 milli of the reagent, 1 milli of the sample. So I add it to the glucose, and usually this is the iodine will give me an instant color change. No need to heat, no need to reduction. This is the egg white, and this is the negative control. I'll try to hold them all together. So as you can see, no change to glucose, no change to egg white, and of course this is my negative control. This is the starch solution. Now the starch precipitate over here, so I'll just give it a mix for you to look at it. Because it's very heavy, so this is the black blue coloration that change when you add iodine to starch. It reacts with it. This is why it will give me a different chemical color change. Our third experiment is testing the egg white, 40% of a protein,
by using the reagent Burette over here. Now I tried as much as I can to show you the original color of the reagent to see the new coloration that I'm having. So this is the glucose, starch, egg white, and my negative control. Now this is art, my students, to hold them all together. I'm just kidding. Now one full withdrawal to the glucose, to the starch, to the egg white, to the water, just a second. Now when you look at the glucose, no change, stays blue. I will just show it to you in a white background in a minute, just a second. Hold your thoughts. See? It's very light to blue, no change. This is for the glucose. This is for the starch. Also no change. This is my negative control, and this is my egg white. Look at it, purplish color. So that is for sure contains protein, the egg white, and it's very important. This is why you see bodybuilders, they consume a lot of eggs to build up their muscles, because it contains protein. Now, as for the fourth experiment, which is a brown paper bag, this is a take-home experiment, which you will use 2 brown paper bag, very small pieces. You'll add a drop of oil and a drop of water to each one of them. Instantly, you will write what you observed, and then you will wait for 30 minutes, and then you write your observation. This is a take-home experiment. Now, from where do you get the brown paper bag? Very simple, Starbucks. Any brown paper bag that you find, the regular bag, just take 2 pieces and do the experiment. You will have to include those results that I'm actually doing in front of you, along with the brown paper bag in the table that you have in your soft copy. And you will also include that table in your result in your lab report. So basically you have the procedure listed in the soft copy, and you have the result. All what you have to do to build up your lab report is to write the introduction and conclusion. Good luck.